as well as the spores while dealing with the palynology. You all know spores are being produced by the plants like thalophytes, bryophytes, and up to pteridophytes. The plants produce these spores, whereas the higher plants, gymnosperms and as well as angiosperms produce the pollen grains. And spores, particularly in the bryophytes and pteridophytes, they have the aperture towards the proximal side. Aperture is nothing but that's the germination area. And after germination, the bryophytes and pteridophytes bear the archiconia and arthrodia, and after fertilization produce the diploid zygote. Whereas pollen grains, male gametophyte, and the main aim of this is to fertilize the ovule after pollination and fertilization. And spores, mostly they have the monolith and twilight part. This is extended towards the proximal side, whereas pollen grains, they have the very type of apertures like colpus, porous, or combined apertures called colporate or pororate, etc. And they are confined to the distal side or equatorial, or they are present all over the grain. That's called omnipresent.
Sir, excuse me, sir.
Yes.
Slides are not moving. Slide share.
You are seeing the sports and pollen, no?
No, sir. First slide is there. What is palynology? It is not moving. No, sir. First slide is there only.
Is it?
Yes, sir. Yes.
Okay.
Slide share చేయండి. అది అంటే పక్కన ఉన్న మిగతా ఇవి కూడా కనిపిస్తుంది. Yes.
Now it is in slide.
PPT.
Is it fine?
Yes, sir. Differences between these slide is there, but the left side, all the other slides are also visible.
Sir, start the slideshow, sir.
Yes,
Slideshow.
I have started from my side. I don't know the technical problem. Actually, I have started the slideshow.
Yes, sir. Just now, second slide was visible now. So only first slide is visible. Second slide difference is between.
Okay, fine. We'll move. No problem.
Okay, sir.
That's not a problem.
All right.
Yeah. And the main difference between the pollen and spore is spores has got a wide ecological range, whereas pollen are restricted to germinate only on the stigmatic surface. That's a major difference. For example, you see, if you keep a bread on a table for 2 days and 3 days, you leave it, then you can see the generation of the fungus on that. That means spores have come from the different places, and they have got the suitable place, and they have started the germination. That's a capacity of the spore. Wherever it gets the suitable media, then it can germinate. Whereas pollen, it has to reach on the stigmatic surface of its own species. Then there it has to germinate. Highly restricted area. That's a major difference between the pollen and the spore. Is it visible? Third slide.
Yes, sir. Okay. Yes, can proceed.
Yeah, this is the answer where you can see this is the Dipticus anther, where you can find the four locket. There are two types of anthers, monophycus and Dipticus. Whenever you see that Dipticus anther, you can see the four locule, and each locule has got microspore mother cells. They undergo division and they produce these pollen grains. Once they get a maturity, this is the place called a stomium, which is a very hygroscopic in nature. From there, it releases the pollen grains out. So, anthers play an important role here. Until maturity, they remain in the anther. Once they get a maturity, Strontium has a key role to release the pollen grains, but all pollen grains may not come out at once. There is a releasing, batch-wise releasing of the pollen grains from the anther lobe to secure the, or to keep the pollen in a viable state. So, can you see the another slide? This is the pollen production and dispersal. Hello?
Yes, sir.
Can you see
Pollen
the?
production is.
Yes, yes.
Yes, sir.
Okay. I don't know if I start the slideshow, it's not moving. Just confirm it, whether it is moving or not. Are they moving? Slides are moving.
Good.
Are they moving? I'm moving the slides. Can you see the movement of slides?
No, sir.
No. Okay, some problem is there, I think. Okay, we'll go with this.
Impossible.
Yeah. One by slide by slide, we will let you know. No problem. That's not a problem.
Yes, sir.
Yeah. Pollen production and dispersal. This is also another important aspect where each plant try to produce a huge amount of pollen because the vectors which takes the pollen for the pollination and the pollen reach onto the stigmatic surface. This all depends on the availability of the vector. If the vectors are not available, within short span of time, pollen loses its viability, and though it reaches onto the stigmatic surface of its own species, they cannot germinate. That's why plants always choose or release a batch wise of pollen grains, that to viable pollen grains out, and the quantity is always matters. They release huge amounts of pollen grains from the anther node so that a few of them can reach the stigmatic surface. And there they can germinate and produce the pollen tube that reach up to the ovule, and there it discharges the sperm cells. One sperm cell go and fuse with the egg cell, and other sperm cell go and fuse with the secondary nucleus. That's all, full fission and double fertilization and all other aspects, embryonic aspects. Okay, so pollen production is always an important aspect. It always depends on it. It is a species specific. Each plant has its own mechanism or its own number of pollen grains per anther they release and reach the stigmatic surface. Basically, there are two types of representation can be seen. One is over-representation, another one is under-representation. Over-representation is nothing but when a plant is not there, and pollen abundance can be seen in that areas because somewhere else the plant is there and it produces a huge amount of pollen because of the wind or some other that has reached the areas. So such type of plant pollen grains are called over-representation of pollen grains. Whereas under-representation, these are absent when the plant is present because it produces very less amount of pollen grain. So that pollen may be dispersed by the air or somewhere, and you cannot see the see the pollen in the atmosphere. So that type of pollen are called as under-representation pollen grains. So there are various types of mechanisms plants choose because pollination is an important aspect. For that, they have to attract the vectors. To attract the vectors, plants also gives different types of advertisements. You know how we are attracted towards advertisements. Plants also give such type of advertisements, and they call the vectors. Some plants choose the birds as a vectors, that's called ornithophilia. Some plants choose the air as a vector, that's anophily, or insects, entomophily, etc. Which goes on, hydrophily, ophiophily, chiropterophily, etc. Whichever the vector, whichever the plant, depends on that relation, the mutualistic understanding between those, and they perform the pollination services, they provide the pollination services. For that, plants offer the nectar and pollen to the species, to those vectors. Those are nothing but just like advertisements. and even sometimes they have the SOV petals so that vectors are attracted towards that, and the plant never get the pollination. So here, Haemophilus plants always, because they have chosen the wind as a vector, they are also called as wind-loving plants. And the peculiar mechanism that they have, the anthers are versatile. They can move freely because the filament is jointed at the center of the anther, and so that anthers can move freely, lobes can move freely. That's called versatile. The arrangement is called versatile. So according to the direction of the wind, they release the pollen grains to the atmosphere. So wind can take this pollen and reach onto the stigmatic surface. And because of that, depending on the wind, they have to produce more of the pollen. Because sometimes there is a wind velocity is more, sometimes wind velocity is low. Even the direction of the wind also always changes. So there is a chance of reaching the pollen grains on the stigmatic surface, or there won't be any chance of reaching. That's why, to avoid the risk of that not having the pollination, they always release a huge amount of the pollen so that any few of them go and reach on the stigmatic surface and fertilize the ovum. So that's why we are seeing the more production of the pollen grains in the annual plants or wind-loving plants. Okay. For example, if you see a pinus, this is the number per annum, or else even rumex also. Huge number. If you go to the any maize field, whole crop is covered with the during the time of anthesis. You see, if you observe, all plants and even ground also is covered with the pollen dust. Many of the grasses they produce more amount of the pollen grains. So dispersal is mainly because of the air. Air has to disperse this. That's why they always release huge amounts. And distance also. Sometimes they cross the continents also. Pollen cross the continents also. But such distances they travel along with the air. See here, these are the gymnospermous plants. They're releasing the pollen. This is called the yellow rain. Once they release the pollen during the anthesis time, if you observe, just like a cloudy mass of the pollen grains can be seen near to the plants. Such a great number of pollen grains are being released by the Anopheles plants. And one more important thing is Anopheles plants always, the morphology of the pollen grains of the Anopheles plants is always unique because they have to produce the pollen that must be lighter in weight so that the wind can easily carry, dry in nature so that they cannot stick to any obstructing places or any uh, uh, any obstacle which is in between the wind flow, so that they can re-relate to the air and that they can travel along with the air. And also smooth pollen grains. That's called silent pollen grains. No spines, no ornamentation we will see. So that these all helps the pollen grains to reach far areas or to reach the targeted areas along with the air. So, and another thing is high surface to volume ratio. And particularly if you see in gymnosperm pollen, they have the wings also. Pollen grains also having wings. The wing is called as saccus here, monosaccade, bisaccade, trisaccade. That wing help pollen reach to the far distances. So these are all adaptations, pollen morphological adaptations that have changed according to the they have chosen the vector. Here the vector is A. So according to the that vector type, they have got such type of modifications, and finally their target can be reached by these adaptations. And coming to the next one, that's called Jubilous pollen grains or animal living plants. Animal living plants, there are different types. Already I told you. If it is insect enterophily, or snakes, that's ophiophily. Bats, that's a chiropterophily. Or else birds, that's ornithophily. Whatever the vector it is. And here the production is a little low when compared to the anopheles plants. Uh, if you have any doubts, you can raise, no problem in between. Animal loving plants always produce a less amount of pollen grains, because here the vector is directly involving, coming to the flower and take the pollen and directly go to the stigmas of same species or whatever. They go and they pollinate the same species. That's why they don't need to produce a huge amount of pollen grains. So we can see a less amount of pollen grains when compared to the anopheles plants. And particularly here, there's target is target can be reached by the birds or animals, whatever vector they directly can reach with this. So here they don't have to waste their energy in the production of pollen in a higher number. And see, flax produce only 20,000 grains per year. That's a very poor number. And dispersal also. Uh, uh, Most of the time, if you see bees, honey bees, they'll what you call honeycomb, or wherever it lays that honeycomb, it attaches to the honeycomb, it builds honeycomb to the particular plant. From that plant to within the radius of the 3 kilometers or 4 kilometers, maximum radius is a 5 kilometers range. Within that circle the room, and they collect the pollen and nectar. So the radius is also very low. They cannot reach far areas. Maximum distance is 5 kilometers or few meters from the plant. But you can see more abundance of the pollen near to the plants. And they always dispersed in clumps because they have the various adaptations, pollen morphological variations, like they are having the spines and they are having the special term we use, that's a pollen kit, which is nothing but it is a sticky substances, so that all in clumps, and they can attach to the bodies of the insects or birds or butterflies, and they can reach the targeted areas. Nothing but the stigma. And even sometimes the all pollen grains all united or aggregated into the octads, tetrads, octads, and even polyads also. 24 pollen grains, 36 pollen grains, even 60 pollen grains also united a single land mass, single pollen mass, and reach the targeted areas. Even sometimes you can see mass pollen grains in the form of the Palynology, where you can see Asclepidaceae or Hussainialam members. Particularly in Asclepidaceae members, you can see, even Archidaceae members also, you can see this type of pollen masses. They are in the form of Palynology. Here we see this is the bee. You can see where how the pollen are attached to the bodies of the bees. And while moving, while roaming, while collecting the nectar, or else while collecting the pollen, generally bees collect the pollen and nectar. Nectar is the carbohydrate source, sugar source. They satisfy the carbohydrates by sucking the nectar. And to build the body, they require proteins, and the protein source is the source they get from the pollen grains. After they deliberately collect the pollen, and while collecting the pollen, while collecting the nectar, the pollen which are attached to the body parts of these insects just fall on the stigmatic surface, and there they start germination and produce the pollen tube and fertilize the ovule. So here, this slide you can see the pollen accumulation rates among the vegetation types. So Pine forest, it's so high, number is so high. But this is the grains centimeters per year, the fall of the pollen grains at, near to the plants. And deciduous plants, deciduous forest, it has such a range, and the steppe grasslands and tundras, even desert plants you see, cacti, 50 to 20,000 centimeters, that much number of pollen grains produced near to the plant. So there is a difference between the pine forest and the desert plants. So gymnosperms always produce the higher and higher amounts of the pollen grains because their topographical distribution is unique. They're confined to only the high elevated areas or temperate zones of the world, and from there, once they release the pollen grains, conditions are very cool, so they take moisture and become heavy, and most of the time they settle down there itself. That's why releasing more and more of amounts and that to saccade type of pollen grains, they help the plant to reach the or disperse the pollen to certain areas and can get the pollination. So here, coming to the pollen grain, if you concentrate the pollen, uh, you can see two important things of the pollen grain. One is outermost layer, and other one is innermost layer. The outermost layer is called as an exit, and innermost layer is called as an indent. The view of the view of the embryologist or a view of the commentologist, a view of the cytologist, a view of the uh, palynologist is quite different. Embryologist view is always on germination aspects of the pollen grain. We want to know the pollen, how it is generating, from where it is generating, how many pollen tubes is extending, and all these, the length of the pollen tube, and germination aspects, particularly concentrates. That's the interest of an embryologist. Whereas the cytologist interest is only in the inner contents of the pollen grain, the karyotypic or the whatever the inner contents, psychological contents of the pollen grain is a more important aspect for the cytologist. Whereas a palynologist interest is only on the exine surface, exine surface. That's why before going to the study of the pollen morphology, we have to remove all inner contents so that once the inner contents are removed, only the shell part, the outermost layer, the exile part is only left over, so that we can clearly see the morphological characters of the pollen grain. So this can be done by using the isotolysis technique. That isotolysis technique is nothing but where we can add 9 parts of the acetic anhydride and 1 part of the concentrated sulfuric acid. That mixture is called as isotolysis mixture. Once you add that mixture and put it for a water bath for 5 to 10 minutes, and that digests the all inner contents, and it gets the turns the color of the exile turns to the golden brown color. So that is optimum color. Whenever you find a pollen grain or a spore in the form of fossil, that fossil pollen or spores are also look similar color. They do have similar colors. That's why this is called as artificial fossilization. You all know what is the fossilization. Fossilization is nothing but the permanent preservation of the plant parts. It's not a simple thing. It's by not by choice, it's by chance. Very few of them get fossilized, and that's a permanent preservation. If any excavations reveal that pollen or spore, if you observe on a microscope, their exine color is golden brown color. So here, artificially we are getting that golden brown color doing the electrolysis process. That this process is also known as artificial fossilization of the pollen grains. And one more thing, we are removing the inner contents, and so that we can concentrate purely on the external features of the pollen grain. Then we can come to know the variations between the variety of pollen grains. Uh, Here, the morphology. Why to study the morphology? Pollen morphology. What's the importance of pollen morphology? Of course, we can utilize this pollen morphology in many ways, but one important aspect of the applied aspect of the palynology is palynotaxonomy. Taxonomy is nothing but it has three aspects, major aspects. One is identification, second one is nomenclature, third one is classification. Once you identify the species, then you have to give a name, then you have to classify that species. So identification can be done based on the morphological characters, plant morphology, or anatomy, or embryology, or cytology, or even you can rely on the palynology. The pollen morphological characters can be utilized in the identification of the plant. That's why it has got a great importance in the taxonomy. So this study is application of this plant's pollen grain morphology in the identification of plants is known as palynotaxonomy. Dr. Abraz has come out with one book that's pollen flora for Nizamabad district. That is only the book available in entire India. Earlier, very long back, Nair came out with one pollen morphology of pollen flora for Maharashtra, very long back, 1960s. And till today, there is no such book. And we have come out with that book along with Girraj. That's pollen flora of Nizamabad. This is only the book available in India. And plant taxonomy, taxonomy is also has become a very important tool in the APG, angiosperm phylogeny group of classification. So here, why to study the morphology? We have seen because morphological variations are there, and these morphological variations can be used to identify the plants or segregate the plants. So, to arrive at any relationships between the pollen morphology and taxonomy, it is very important that pollen material should be collected from authentically identified material that's called either holotype or isotype. You all know typification, ICN, International Code of Nomenclature, Article 7 talks about the typification type methods, where holotype is nothing but the type of specimen which is used by the author at the time of description of the new species. We rely on that and they describe the new species based on that specimen, a specimen, and he deliberately mentioned that specimen as a holotype. So that is a holotype preserved. Such type of holotypes and isotypes are preserved in any one of the recognized herbaria. Here we do have one international recognized herbaria. There is in short form it is named as HY, Herbarium Hyderabad. Hussainialam University Department of Botany is having such a herbarium, and more than 10,000 plant specimens, herbarium specimens are preserved here. Dr. Abraz sir collections also preserved here. Okay, so most of the time holotypes are not allowed for a collection of such a material. Isotypes you can, because isotypes are more. Any one of isotypes you can take, so that you are going for an authenticated specimen. You are taking the pollen from the authenticated specimen, and you can go for electrolysis and you can identify the plants. So why such a variation you find in plants? See, these morphological variations, pollen morphological variations, are always because, for example, can you see the slide here? Y pollen variation slide. Can you see here? Is it
Yes, sir. Okay, sir.
Yeah.
Yes, sir. Yes.
Yeah. See, once the plant come to the reproductive stage, flowers once they blossom, they open, and the stigmas are open to the air. They put stigmas open to the air, and now the vectors, whatever the vector it is, if the plant particularly choose and bees as the vector, and the bees comes and visit the flower, not only the Mangifera indica flower, just imagine it's a Mangifera indica flower. It is open, and the bees visit various type of flowers, Tamarindus indica, Azadirachta indica, whatever, many flowers they visit, and they also comes along with the all other plant pollen to the other Mangifera indica flower. And here the stigmas, because they are open to the air and they are accessible to the vectors, and they come along with the different types of pollen grains, and they put all the pollen onto the stigmatic surface. But here, stigma plays a crucial role, very important role in the identification of its own pollen. It never allows other pollen for germination. Now, other pollen also can reach out to the stigmatic surface. Stigma identifies its own pollen and allow only that pollen for germination, and pollen tube of that such pollen grain comes along with the style and reach the ovule. There it discharges the sperm cells. That's the beauty of the system here. For this identification, each and every plant go with a different unique type of ornamented pollen grains, patterned pollen grains, size variation, shape variation, etc. etc. And even because of based on the vectors also, there is a variation. So that plants always release such type of varied type of pollen grains, and stigmas of such plants can recognize, identify such pollen grains on the stigmatic surface once they turn it over, and they allow them for a germination, and they get a pollination and a fertilization. Okay, so this variation we can utilize in the identification part. That's one of the important aspect of taxonomy. And here, that morphological characters, the variations of the pollen grains can be categorized into these things. One is the pollen units, other one is polarity, other one is size and shape, symmetry, apertures, and extent stratification. So coming to the pollen units, I told you in the beginning, the stomia, once the maturity, they get a maturity, anther get to maturity, pollen grains, because due to the meiosis, meiotic divisions, each meiotic division leads to the formation of 4 cells, and after maturity, either they release out from the anther group by separating from the tetrad as a single or by combination of 2, or by combination of 4 as these come out, or else double number of the 4 cells they divide further and they release out from the anther lobe. It all depends. So they are called as pollen units. If a single pollen grain gets separated from the tetrad and released out from the anther lobe, then they are called as monads. 2 combined and released out from the anther lobe, they are called as dyads, tetrads, octads, etc. etc. Polynia even. Okay, because of shortage of time, I'm not going in detail. It's already, I think, 1 o'clock. What's the time left over for me? Dr. Gas? What's the leftover time for me? Yes, no one is there.
Sir?
Madam, Latha madam.
Sir?
What's the time left over for me? Can I take up to 1:15? Can I explain?
Yes, sir. You can.
Yeah.
Sir. Okay, sir.
Okay.
Thank you.
So here you see in a family Mimosaceae. Mimosaceae. Can you see the slide? Mimosaceae.
Yes, sir. Yes, sir.
Yeah. Mimosaceae. We can identify the pollen grains based on their pollen units. For example, Prosopis julifera or Prosopis specigera, Jambi and Sarcopuma, they release pollen grains from the anther lobes in the form of a single unit. They are called monads. Whereas the Mimosa pudica, this is a tetrad pollen, and Mimosa rubicalis it produces the octads, 8 pollen grains unit and comes out. Whereas Acacia nilotica, Albizia lebeca, these are all polyads, pollen grains released out from the anther lobe. So within the family there is a variation. So based on that variation, we can segregate the plants and we can identify the plants. This is belongs to the so and so species. Okay. So polarities also matters, always matters, and there are two types of grains. Generally, once they are formed after meiosis, four cells are formed. All these four cells, once they are united in the tetrad, in the tetrad form, we can know the direction. Yes, 5 minutes. Yeah. So in tetrad, there are two poles for each pollen grain. Just you see, this is a tetrad. The surface, the face towards the center of the tetrad is called as a proximal side, or else that's also called as a proximal pole. And the center, away from the place away from the center, this place is called as a distal pole. So for this pollen grain, this is the proximal pole and this is the distal pole. For this is the proximal pole, or this is the distal pole. Like this, we can identify the poles, two poles. Each pollen has two poles. One is the distal pole, and another one is the proximal pole. I told you in the gymnosperms, apertures are towards the proximal end, whereas in angiosperms, either it is on equator or distal or all over the pollen grain. That also makes some difference. And the imaginary line which passes through these two polar regions is called as a polar axis, and the other line which is perpendicular to the polar axis, that is called as an equatorial line. So pollen apertures are fall on the equator, then they are called equatorial apertures. And the poles, the distal pole and the proximal pole, this is a line which passes through these two is called as a polar axis, and the perpendicular to the polar axis, the line is called as equatorial axis, and the yes, any doubt? Yeah. So the measurement of the polar axis length and the diameter of the equatorial axis, if you measure these two, and you will come to know the shape of the pollen grain also. So based on the polarity, pollen grains are categorized into isopolar. Both poles look similar, apertural wise or augmentation wise. Then they are called as isopolar. Heteropolar, one side aperture is there and the other side aperture is not present. Or the one side they have one type of ornamentation, other side ornamentation is absent. Such differences you find at the polar regions. There is a difference between the poles. Then that type of pollen is called as heteropolar. And apolar, you cannot make out the difference because of the shape of the pollen. Then that type of pollen are called as apolar pollen grains. And symmetry also matters many times. And either they have the two types of symmetry generally, radial symmetry or bilateral symmetry. Radial symmetry, if you cut the pollen grain from any plane, gives the two equal halves. That's called radial symmetry. Most of the angiospermic pollen are in radial symmetry. Whereas bilateral symmetry means only two planes gives two equal parts, other plane it cannot. Then that's called as a bilateral symmetry pollen grain. Size, there is a much variation between the size of the pollen grains. Even communities, if you see family, or else any family you see within the species, within one genus, the species, part of that species. For example, Terminalia arjuna, Terminalia bellarica, Terminalia catapa, Terminalia mandali, Terminalia arjuna, Terminalia tomentosa. These are all species that can be segregated only with the help of the size patterns. Otherwise, other morphological features are similar, but there is a little deviation from the size. So size always matters in the segregation or identification of the plants. So here, generally, the size of the pollen grains varies from the very small to the gigantic size. Very small size pollen grains are measured between or less than the 10 millimicrons, whereas the small ones between the 10 to 24 millimicrons. Medium sized are 25 to 49 millimicrons. Large pollen grains diameter varies from the range from the 50 to 99 millimicrons. Very large are 100 to 200 millimicrons. Whereas more than 200 millimicrons, then they are called as gigantic pollen grains. Generally, the gigantic pollen grains are seen in the Malvaceae and as well as the Convolvulaceae families. Very small pollen grains can be seen in the Ebanaceae family, Puntinja calabura. The plant is having very small sized pollen grains. And shape, this is also very important. Shape also gives you some clues in for the identification of plants. And these are all various types of shapes can be seen, can come across the pollen morphology. These variations, size variations, shape variations, circular, elliptic, angular, and again under that again different shapes. Angular, triangular, even triangular. The this is sides are concave, concave triangular, straight triangular, or else a convex. Then that's a convex triangular. These are all various shapes. Generally, the shape of the pollen grain is measured and confirmed by the measuring of the length of the polar axis and the equatorial diameter. Based on that, we get by measuring this and we get some ratios. That ratios fall less than 0.50, then that's called paraoblate. Generally, spherical shape, spherical to we go minus if the length of this polar axis, the equatorial diameter length increases and the polar axis length decreases, it falls under the oblate. If the polar axis length increases and the equatorial diameter length decreases, then they fall under the prolate. So based on the variations, size, again they are subdivided into suboblate, oblate, paraoblate, and again here also subprolate, prolate, and petprolate. So this is a very important formula where we use to segregate the pollen grains based on their shapes. So here you can see paraoblate, oblate, suboblate. So their PA by ED 100 values are less than 50, then they are paraoblate, 50 to 75 oblate, 75 to 88 suboblate, and 88 to 99 they are oblate spheroidal. If that ratio comes exactly 100, then that is a spherical round box. So here you see, here polar axis length is long, more, whereas here the equatorial axis length is more. If the equatorial axis length is more, the shape comes under the oblate. Polar axis length is more, that comes under the prolate. So again, differences leads you to confirm whether it is a paraoblate or oblate spheroidal or a prolate, perprolate, prolate spheroidal, etc. So this is a prolate one, this is the oblate one, this is the spheroidal one, basic shapes. So here you see, this is oblate, again prolate, and this is a spheroidal. Most of the Acanthaceae members can be seen in a prolate form. So aperture is also another important thing where we depend for identification of the plants. So here aperture is nothing but it is a open thin area which is present on the surface of the exine. While formation of the exine, it leaves some gaps. Those gaps or thin areas are called as a aperture. So these apertures are meant for the germination of the pollen grain. Because there is no direct contact between the male and female gametophytes in plants. Because the female gametophyte is embedded inside the that ovary, and there is a style, long style, and there is a stigma. So pollen has to come out of the stigmatic surface. There, it has to produce the pollen tube. That pollen tube elongates and reach the female gametophyte. As long as the style is, that much length of the pollen tube must be produced by the pollen. So you can see very long styles in the that maize, Makuta, that whatever you see that whatever you call it as a pichu, that's nothing but they are all styles. So very lengthy styles you can see in maize. So that much length of the pollen tube also they produce. Okay, so if the grains, pollen grains are having the round aperture, round opening, thin area, then that's called as a pore. And extended aperture, that's called boat shape aperture, is called as a bulbous. If the both are combined, then that's called as a pulporate. Uh, so these are all various types of apertures can be seen in pollen grains. In aperturate, where you don't find aperture, but it is also called as omni-aperturate condition. That means once a pollen landed over the stigmatic surface, from any part, any area, any surface, it can produce the pollen tube. That condition is called omni-aperturate condition or inaperturate condition. So that it has that advantage. Okay, leptomate, pulpus, spore, etc., etc. These are all. And there is a classification called NPC system of classification that was introduced by the Earthman in the year 1961. So this is very important classification. Classification based on the aperture patterns and placement of aperture. N represents the number of apertures. P represents their position, and C represents the what type of aperture it is, the character of that aperture. So based on that, he has come out with the varied things. So here you see, this is the number. Based on the number, N1, N2, N3 goes on. If one aperture is present, that's a monoaperturate. Two are present, diaperturate, triaperturate, tetra, penta, hexa, polyaperturate condition. Whereas kata, that is present towards the proximal side, that's a kata. Proximal and distal side, then that's anakata. If it is confined to the distal, then that's ana. They are distributed on the equatorial line, then that's zono. Both two rows of apertures can be seen on the equatorial line, that's dizono, and they are distributed all over the grain, that's anto. And type of aperture, trichotomo, pulpate, boat shape aperture, round aperture, that's a porate, both combined pulpus and porate, that's pulporate, both pore and pore within the pore, that's a pororate, etc. So 3-digit number we get with the result of this NPC classification. Based on that 3 digits, we can confirm the type of pollen. So these are the merits of this NPC classification. It is a simple system of classification and illustrates the apertures of the pollen grains and the spores. Very simple classification system. Just like artificial classification system, lineage, you can relay this system. You can study this system under the artificial classification system along with the lineage classification system. That's a sexual classification system based on the number of stamens. He has come out with that, introduced that system. So these are all merits of this, and of course, because of time constraint, I'm not going in detail. These are the demerits. This is an artificial system of classification. So syncopate and parasyncopate pollen grains do not fit neatly in NPC classification system. Of course, there are definitely any system has its own advantages, merits, and demerits. This to have merits and demerits. So this is showing this slide is showing the early dichotomies and monoquaternions, how the distribution patterns of the pollen grains aperture in the that is evolved in different plants of the dicots and different plants of the monocots. So monosulcate aperture, this is proximal side aperture is transformed, evolved into the this type of apertural patterns, ulcerate and non-aperturate and tricolpate aperture. Like this, panthophorate aperture. Like this, there is an evolution of the apertures, apertural patterns, so that you can see this variation patterns in plants. And this is all about the monocotyledons. Here again it is a monosulcate. Monosulcate leads to the formation of all these types, and this greatly helps in the identification of the plants and segregation of the plant groups also. And the main role of the aperture is actually we are utilizing the aperture pattern in the identification of plants, but the main function of the aperture is for germination. And also another one is the term we use, we call it as harmomegathi. Harmomegathi is nothing but once the pollen grain is placed on the stigmatic surface, stigma have the exudates, sticky substances, sugary substances, exudates. Once it is landed on the stigmatic surface, because of that exudates, first step for the germination of pollen grain is hydration of the pollen grain. It absorbs the moisture. So it absorbs more and more moisture or water because according to higher concentration to lower concentration on this moves. And there's a movement of water more and more, and that leads to the increasing the size of the pollen grain. Ultimately, sometimes there is a chance of the bursting of the pollen grains because more excess amount of water penetrates into that, imbibes into that pollen grain, and there is a chance of bursting. To avoid that such bursting, the pollen, that apertures gives the elasticity to that pollen grain. So that elasticity nature of the pollen grain is called as a harmomegathi. Coming to the another important aspect of the morphology, that's called pollen exine structure or exine ornamentation. And this exine ornamentation also varies plant to plant, and this greatly helps in the identification of the plants. So here basically this has two types. One is an exine and other one is a sexine. Exine has exine is divided into sexine and a nexine. Nexine is nothing but a non-sculptured exine is called as a nexine. Sculptured exine is called as a sexine. Here S represents a sculpture. Sculpture means various patterns, ornamentations. So those ornamented patterns part is called as a sexine. Non-ornamented part is called as a nexine. So nexine is having the ectexine and endexine. This is called intine, and endexine. Above this, you can see the fruit layer, and above this, you can see rod-like structures. They are called as a polymella. Above this, there is a roof layer called as a tectum. If the tectum is absent, ejected, tectum is present, then that pollen grains are called as a tectate pollen grains. So if tectum is absent, then the rod-like structures are present. They are called as a vacuole. If tectum is present, rod-like structures are called as a polymellae. So this is a Thackeray and Iverson's view is different, and Edmund's view is different. But based on that, you are seeing so many terms are here to explain the structure of the exine. But don't get a confusion. It's very simple. Two terms you can use nexine and sexine. Nexine is nothing but a non-sculptured part is called a nexine. Sculptured part is called as a sexine. Simple. So above the tectum, if you find any ornamentation patterns, that's called as a supratectal ornamentation. Below the tectum, if you find such ornamentation patterns because of vacuole arranged into different patterns, then that's called as a subtectal ornamentation patterns. These are all greatly helps in solving the taxonomical problems. And so this is what we are discussing about. And there is a much difference between the exile ornamentation patterns of the gymnosperms, pteridophytes, gymnosperms, and angiosperms. So in pteridophytes, there is a simple structure, that is a laminated structure, or in some pteridophytes, along with the laminated structure, you can see the undulate patterns. This is called a parent. And whereas coming to gymnosperms, there is a much evolution, much development, and along with this laminated structure, you can see the cushion-like patterns that are called as alveolar structure. Presence of the alveolar structure along with the laminated layer is an important character for the gymnosperms program. Whereas exile structure of the uh, angiosperms is much diversified, much classified, and already discussed the various patterns of that endexine, ectexine, and foot layer, columella, tectum, and the supratectal ornamentation, subtectal ornamentation, etc. etc. So in gymnosperms, coniferals, you can see the Pinaceae members has two types of pollen grains. Winged pollen grains can be seen in the Pinal, Girraj, Picea, these. Whereas wingless pollen grains, Larynx, Suga, Arcaria, and Agathis. In Taxaceae, Doria and Taxus show the annulate or orbiculate pollen grains, whereas the Photocarpus they have the bisectate or bisectate annulate without vacuoles. So, details again it contains the three types of genera, Nitum, Ephedra, and Welwitschia. Nitum pollen grains are non-aperturate, no apertures are seen. That's another term is also I told you that's the omni-aperturate condition, and spinulate, spinules are present. Whereas Ephedra it has 5 to 15 ridges and grooves without any permanent apertures. Just grooves, ridges and grooves pattern can be seen in Ephedra. Whereas Welwitschia, monocolpate, see the important character here, monocolpate. Monocolpate condition is also another important character for the monopods. Monopods. That's why there is a changed pattern, monophyly, biphyly, polyphyly evolution. So to establish the monophyly, APG classification, you are seeing the monopods have come very closer to the early angiosperms. Even this palynological evidence also supports such type of things. And angiosperms have got a very varied type of conditions, patterns. So here you see these are the various patterns. We place them in the identification of angiosperm of species. So ciliate, if the tectum is smooth, no ornamentation, that's a ciliate. Scapate, some ornamentation. The verrucate, have round structures, all above the exile. Then that's called verrucate ornamentation. Reticulate patterns, that's a supratectal ornamentation, subtectal ornamentation generally. The baculæ arranged like this and forms the reticulum form. That's called reticulate ornamentation. Fabulate, perforate, perforations, equinate, striate, etc. etc. These are all. And basically, pollen grains are divided into two categories. One is a stenopilness. This is also called as a unipilness condition. Throughout the family, pollen morphology is uniform. So such type of families comes under the stenopilness condition, or they are also called as unipilness families, like Asclepiaceae, Brassicaceae, Poaceae, Lamiaceae. In Poaceae, you cannot segregate them just by based on the pollen morphological patterns, except the size of the pollen grains. All other characters are very similar. Silent ornamentation with a monoporate condition. Unipilnous condition, there is a variation plant to plant within this genus. The species, each species has its own type of ornamentation patterns. So that's called the unipilnous condition. That is also called as a multipilnous condition. You can see this, you can come across the families like Astraceae, Acanthaceae, Rubiaceae, Botany, etc. This is because of the heterozygous patterns. They form the various patterns, and that patterns helps you in identification process. This is all about the pollen morphology, what we have done. And other important applications of the palynology, if you see, there are many important applications with this. Also, palynology is one important aspect where honey samples, you can confirm the purity of the honey based on this pollen analysis. If you find pollen of different plants, you can confirm the you can confirm the geographical source and as well as the botanical source. If you give me 10 ml of honey, I can identify the source of the plants where the bees have visited. Each and every plant they visited and they collected the nectar. Those plants can be identified just by analyzing the honey sample at the laboratory. We can assess, we can analyze the plants which were visited by the bees. And we can also know the geographical source. For example, in advertisements you can see lion honey. In tagline it is called as Kashmir honey. Kashmir honey means Cucus sativus. That's a kunkuma one. Cucus sativus pollen represents that pollen in the honey samples. Then we can confirm that these samples have come from the Kashmir locality. Like this, we can segregate Ooty honey, Kurk honey. For example, Strobilanthus kulpiana. That plant comes to the flowering once every 12 years. Every 12 years, only once comes to the flowering. Once it is in flowering, all mountains are looks blue. That's why they are called the Nilagiris. Nilgiri honey can be detected presence of this Strobilanthus kulpiana pollen grains. Like this, we can identify the botanical source as well as the geographical source and also purity of the honey samples. We can confirm. And clinical palynology, this is another important palynology where we can study the allergies. Generally, pollen is coated with the various proteins, and that proteins, once we inhale, our immune system reacts immediately because the foreign proteins are entered into our body, our system. Our basal membranes recognize it and try to combat with this, fight with that pollen proteins. And during that time, the mast cells, which are innocent cells, get bursted and release the histamines. Those histamines are positive for the getting allergies. So some people are very sensitive, very susceptible to the pollen grains. So far, many of us are not aware of the having such type of allergies, but we are the sufferers of that allergy. If you are getting every year in same season, continuously any cold or a cough, you are allergic to such type of pollen grains. But we are not aware of that. We think that it is because of the virus, or we may think that it is a flu, normal flu. We take medicines of the flu, but many times it is because of the pollen. If you neglect that allergies continuously, persistent allergies you are prone to, then slowly that will turn to the asthma. Once the asthma is triggered, that's not curable. You know how the asthmatic patients suffer. They suffer a lot. Particularly the Palynology, the plant Hyderabad, is also called as a Congress weed. It has entered into the India while importing the wheat from America. Now it is a part and parcel of this, and it has it cause such type of allergies. And forensic palynology, where we can utilize the pollen grains in solving of the various crime or detection of the crime. So pollen has such capacity because the pollen once the exine is coated, the outermost layer exine is made up of sporopollen. It's a very tough and rigid layer which cannot be destroyed easily by the physical, chemical and biological substances. So it can remain for a long time, even in the fossil form also it can available. So those pollen can be utilized in the solving of the crime or detection of the crime. That comes under the forensic palynology. So paleopalynology, where I can study, excavate the different places, and from there you can recover the pollen grains, and such pollen grains, fossil pollen grains, based on that, we can reconstruct the past vegetation patterns, what type of vegetation was there in such localities, and based on the past vegetation patterns, we can predict the past environmental conditions, what type of environment was there, climatic conditions, past paleoclimatic conditions were there. So this is also another important aspect. Thanks to the Svante Peppo, who got the Nobel Prize recently in medicine, who worked on the paleogenetics. Again, everyone looking towards the fossil aspects and all. So paleopalynology is also having such a interesting thing, and even we can identify the availability of the what you call the natural gas and petroleum. These are all okay. So hydrocarbon exploration, particularly petroleum and all these, to identify these. Pelon is also one important tool that's called a TAI index, thermal alteration index, that gives the confirmation of the hydrocarbons in particular locality. And pharmacopalynology, where they can utilize the palynology in the preparation of various medicines, pharma industry, that is called pharmacopalynology. And copropalynology, the excretions, particularly animal excretions or bird excretions, even human viscera excretions. These are all contains the pollen grains, and analysis of these pollen grains and utilizing these in solving of the crime or somewhere else, or reporting something, and all these comes under the copropalynology. And the paleotaxonomy, already we discussed the entire thing, morphology based study or identification of the plants based on the pollen morphology is called as a palynotaxonomy. So these are all important applied aspects of the palynology. So thank you all for patience listening, and if you have any doubts, you can over to our members. Thank you once again.
So, students, if you have any doubt, you can ask. Sir, sir is ready to clarify all your doubts. Now, I request Raviraj sir to give the vote of thanks.
Yes, very good afternoon, sir. My voice is clear, sir.
Yes.
Yeah.
I think students will raise their hands, or otherwise you will unmute your mic and ask any doubts or questions. Anybody, students? Yes, anyone? If there is no questions, we will happy. Deepak sir. If there is no questions, I will proceed.
Sir?
Yeah, it's up to 1:30. You can wind up, no problem.
Yes. Thank you very much, sir, for giving an enthusiastic, elaborative, and very extensive lecture. It covered all the things of palynology. So there's a lot of scope in this palynology, but the students and other people are not recognizing the importance of palynology. So forensic palynology is an excellent branch which has more scope when all the evidences by police personnel which are they are unable to prove the crime scene and culprits. In New Zealand as well as Australia and in England, this forensic palynology helped to solve so many unriddled cases, and they caught the culprits who made some murders. With the help of this Palynology only, those cases are solved. And the Palynology is also one of the important branch in which we have more scope. And recent classifications also proved that APG classification also proved the importance of the Palynology. So continuously uninterrupted, more than 1.5 hour, the big lecture was delivered by our Vijayabhaskar Reddy sir. Thank you very much, sir. With the inspiration of Vijayabhaskar Reddy sir, I chosen my PhD work as a taxonomical studies of reserve forest. We are very fortunate in our post graduation. We heard the lectures of the great personality, Professor C.G.K. Ramanujam. He is an Indian paleobotanist. It is a synonym for gymnosperms studies, and is a great teacher. We are very fortunate. My supervisor Vijay Bhaskar Reddy sir and myself also very lucky to listen his class. And he clearly comes to the his own collection posited, and for the sake of student requirement, so he established the Palynology Paleobotany laboratory. And Vijay Bhaskar Reddy sir also opened the Applied Palynology Research Laboratory at the Nizamabad College. And under sir's able guidance, I was assigned other more work. Definitely, I will complete my Palynology work, sir. And I will insist and guide the students also who are interested on their own. If we enter into the field of pollen grains, beautiful, not like the morphology of taxonomical plant species, like beautiful flowers. Pollen grains are very, very, very beautiful and different, very beautiful ornamentations. So once we enter into the subject, we will enjoy a lot. Thank you very much, sir, for giving an opportunity, and for the benefit of our students, delivered an extensive lecture. Thank you very much, sir.
Thank you so much. Bye bye.
I also sincerely say my thanks to Dr. A. Vijay Bhaskar Reddy sir, Assistant Professor of Botany in Hussainialam University. Sir, your association with this college is every year, sir. You deliver, you extend your cooperation for many things here. So we are very thankful for your cooperation, sir. And I hope this cooperation will continue in the coming years also, sir.
Yes, with pleasure.
Thank you very much, sir. And I also thank Latha madam, Dr. P. Latha madam, who has collaborated with our college in conducting this extension lecture. And of course, there are many more programs that we are going to conduct, madam. And I also thank all the students who have participated. Sir, we have arranged, you can see in my bag,
Yes,
we have
yes,
arranged a screen. Okay, that's why you can see the number of participants is less because we have arranged a separate screen for
Yeah,
them
that's
and
great.
all the things. That's why same thing madam also has done, sir.
Okay.
So thank you very much, sir, for your kind cooperation, sir. Okay, thank you, sir.
Okay. See you all. Thank you. Once again.
Thank you. Thank you.